Hello, everyone. Thank you for joining our webinar, Automating Event-Driven Data Analysis with Nextflow and Seqera. My name is Ken Brewer. I'm a Senior Developer Advocate here at Seqera, and I'm joined today by my colleague, Edmund Miller, who's a Bioinformatics Engineer on our scientific development team. Before we get started, a couple of housekeeping items. If you have any questions during the webinar today, please go ahead and submit them in the Q&A box, and we'll answer some of those as they come in, and then we'll go over the remaining ones at the end. And please go ahead and participate in the opening poll shortly, and then we'll also have a closing poll at the end. And lastly, we will be sharing this webinar recording by email to any participants who are attending today. Perfect. Okay, yes. Presenting myself and Edmund. And then for the housekeeping items, use the Q&A. We'll have a few polls, and then we will be sharing the recording out with everyone in a day or two afterwards. So next, just to give a little bit of an agenda about what we'll be doing in today's webinar, I'm going to start by giving a brief overview of Seqera, what is our mission, and what are we working on as a company. I'll then talk a little bit about what is event-driven bioinformatics, what we have in the title of our webinar, and then talk a little bit about open source and Seqera tooling that we're going to be presenting as part of our demo, which will be sort of an end-to-end automation example of how you can take science that you can automate from the instrument all the way through to the analysis. And that will be led by Edmund. And then we'll have a Q&A at the end of the webinar. So to give a little bit of an overview about Seqera and our mission, Seqera was founded by the creators of Nextflow, which was developed by Paulo and Evan in 2013. And Nextflow has become a gold standard tool for reproducible bioinformatics, in large part due to the creation of the independent open source community NF Core, which has been collaborating on putting together best practice pipelines. Seqera, the company, was founded soon afterwards in order to support companies and universities and institutions that are looking to work with Nextflow in a more team environment where you're going to need to have better reproducibility and monitoring and logging of all of the things related to your Nextflow workflows. So fast forward to today, we see over 2 million Nextflow executions per month. Or getting close to the point where Nextflow is being run once a second. And Seqera's commercial platform has over 120 customers, many of the top pharma, lots of mid-sized biotech, and also a number of academic institutions and large governmental organizations like the CDC and Genomics England, and many more. And we see as our mission at Seqera as advancing science for everyone through software. And this just comes from the idea that better software can really enable better science. And the core pillars of how we try to do that as a company, we like to talk about in terms of our tools for helping scientists build, which is mainly Nextflow, MultiQC, and Wave. We have tools to help scientists discover tools and software for doing their analysis, the main ones being Seqera containers and Seqera pipelines. And then finally, tools for helping scientists and organizations scale their science. And our flagship commercial offering is Seqera Platform that we'll talk about in more detail later. And then we also have Fusion, which is a high-performance software for accessing data much more quickly that can accelerate and make your pipelines run faster and cheaper. So let's jump into the main topic of the webinar, which I titled Event-Driven Bioinformatics. And when I think about what is event-driven bioinformatics, I like to contrast that with the alternative, which is something that I've seen in my career. Prior to joining Seqera, I spent some time as one of the first computational hires in a small size therapeutics-focused biotech, trying to discover novel proteins that might be interested to develop into drugs. And I also spent time as a software engineer in a large clinical genetics company building a bioinformatics platform to process hundreds of patient samples a week. And across both of those, I've seen that bioinformatics, or like the flow of scientific data within labs and organizations, often kind of falls into this type of flow, where you have the wet lab that will do experiments and generate data, and then they will pass that data off to the dry lab, the computational people and the bioinformaticians, who will do some sort of like pre-processing, run pipelines, and some initial analysis. And they'll pass off the process data to either like analysts or leadership. And in therapeutics-focused biotech, those analysts and leadership might decide on like what are the next steps to take in terms of other experiments to run. And when you think about the tools that are most often used in human-driven bioinformatics, there's a lot of ad hoc processes, and the data and metadata will flow through tools that are perhaps suboptimal for data integrity. For example, the raw data might come off the instrument and be immediately copied by a scientist into an Excel spreadsheet, which gets uploaded into OneDrive, and then that gets sent to the bioinformatics team, who will then put together some slides in PowerPoint and pass that on to like leadership, who will then perhaps write up a report with like instructions for future experimental directions to do. And all of this is sort of orchestrated with Microsoft Teams or whatever Slack messaging system people use. And this is a system that is highly flexible, but starts to break down when you try to scale to larger volumes of data and a focus on greater reproducibility within your science. So when I think about event-driven bioinformatics, I like to think of that as focusing on automating data handoffs, data processing, and report generation to make them so these processes can happen as smoothly as possible without the need for human intervention as much as possible. And Seqera has both open source and commercial tools that really help a lot in this process. From Nextflow, which can automate a complex multi-step pipeline that a bioinformatician used to run by hand in the terminal into a single command that can be executed in the terminal or in the cloud. And then multi-QC, which works wonders for creating graphs and generating reports that can be just easily tacked on to the end of a pipeline. And Seqera platform works very well as a central orchestrator in all of that. So when I try to break down the processes that you might need to do to create an event-driven bioinformatics workflow, I see 6 key steps. And later in our demo, Edmund is going to showcase how each of these can be accomplished with a combination of 3 pieces of tooling from both open source and from Seqera. And those key processes are uploading the raw data from your instrument. You often need to fetch metadata about the data. So if you have like a sequencer run ID, you'll need to get information about, what type of processing is required for this data. And you'll want to have that travel along with the data as much as possible. You'll need to trigger some sort of scientific workflow. You need to be able to monitor and log workflows that are in process, so you know like when they complete, when the data is ready, and where that can be found. You need to be able to generate reports, stuff like quality, QC stats about the scientific experiment, also the perhaps initial and analytical results. And then you'll often want to explore and do some form of interactive analysis. And all of these things can fit together really well when you can keep them into sort of one cohesive whole. And that takes me to the next topic, which is a little bit of an intro to the open source and Seqera tooling that we'll be including in our end-to-end automation demo. And there are three core pieces of software that we'll be talking about. One is Nextflow, which many of you are likely already familiar with. One is Seqera Platform. And then the next one, which is not a Seqera product, but which is open source, is called Temporal. So for Nextflow, the core idea of Nextflow when it was first developed was to bring modern software development tools to bioinformaticians. And it works really elegantly by connecting your code repositories, different executors that can take the logic of your pipeline and translate them into the commands for running on a Slurm environment, in AWS Batch or Kubernetes or any of the common execution environments that get used. And then automatic parallelization of the workflow to the amount of data that gets put through it. And it really excels at being something that's both portable, reproducible, being polyglot, being able to tie multiple different software pieces together, having both like a CLI interface and just having this unparalleled scalability of taking a single scientific workflow and scaling it out to however much data you can request, instances you can request from AWS or any other cloud execution environment. Beyond that, the other key tool that we'll be talking about is Seqera Platform. And Seqera Platform, the intent is for it to be a unified platform for the entire scientific data analysis lifecycle that allows you to connect your code repositories, manage the infrastructure related to your execution environments, whether it's Slurm, AWS, GCP, or any others, and then work with the foundational open source tools and bring them together with things such as tooling that lets you explore and manage the data, launch the pipelines, analyze the notebooks, and interact with all of these things in a sort of shared workspace with members of your team, and also to have centralized logging, cost control, and a lot of optimizations that can drive down the cost of running your workflows at scale. Now, the last piece of technology that we are incorporating into our solution is called Temporal. And this is an open source durable execution engine. It also deals with workflows, but workflows for Temporal are intended to be something very different than what Nextflow thinks of with a workflow. Temporal was originally developed by engineers at Uber. And so the types of workflows that Temporal is optimized for are things that are asynchronous and intermittent, and that you need a high level of error resilience and advanced retry and recovery logic. And it's very good at tracking the state of processes over time and kind of translating the business logic of the things that you need to do as a business into code. So if you think about like the process of matching an Uber driver to a customer who needs to go from destination A to B, polling different potential drivers to see if any of them want to take the trip, doing the match, and then finally like charging the payment method of the customer, sending a receipt. This is a workflow that's going to not have a strict start and end that can be very tight and where you'll be continually processing that same thing. Instead, you're going to have a lot of starts and stops. And Temporal works really well for sort of buggy, intermittent, and flaky processes. And so our automation demo is going to showcase kind of how you can use Temporal to automate some of the buggy parts of this end-to-end bioinformatics process, and works really elegantly with Nextflow and with Seqera platform. So next, we're going to go to our end-to-end automation demo. And as a reminder of like the six key things that we're looking to do with this is uploading data, fetching metadata, triggering workflow, doing the monitoring and logging, generating reports, and then doing some explore and analysis. And I'm going to pass it over to Edmund, who is going to go through this demo and showcase an example of how you might stitch these things together. And also we'll showcase Seqera platform and how it works as a tooling to support companies in these goals.
Okay. Hello, everyone. I'm Edmund Miller. Previously, I worked at Element Biosciences, and we did a lot of automation through those. I'm now a part of the SciDev team here at Seqera. So I would love to give you all a demo here of the various pieces that we can do with some automation in platform. So first, let's talk about workspaces here. And this is just kind of a full orientation of the platform. So within this, we can have various orgs of them and the various pieces. So this might be your company, this might be various orgs within your larger organization. We have one called Hello Automation here for today's webinar. And then we have a workspace within it called Demo. So this can be within your team. You can have different workspaces for different projects, different sections, whatever you can imagine. So I kind of like to, so I'm going to hop into the different org here. So we have Demo and Hello Automation. And then I kind of like to start from the back of the platform to kind of get everyone orientated to that. So let's first start with participants here. So we have myself, we have the DevRel team, we have the SciDev team, and we can have different role-based access controls in these for various pieces with within the org of just being able to launch, just being able to connect and run data studio sessions, or being able just to view on those. And so then we can also create secrets as well. And then we can hop over here and we can go to workspace credentials as well. And so this we just have is our site of playground. So this might be AWS credentials. This could be SSH keys for your HPC environment. Lots of different options to add those. But here we just have some AWS keys, and these are for the AWS Batch Forge creation of a compute environment on those. And so from that, we can hop into compute environments, which is kind of the next piece. And so we have two different environments here, but I'll just hop into the human-driven bioinformatics compute environment. And this is just AWS Batch by itself here. And so you can see that we can enable various things. We can just use Batch Forge, which actually will just spin up the entirety of the AWS environment for you just from those keys. We can have spot or on-demand instances, and we can have various buckets attached to this compute environment. It's so that's kind of been the back end of platform and the setup for this that's pretty standard across all the orgs. But you may already have an environment in platform and you're just wondering, how do I automate these things? And that's where we're more here today. So let's talk a moment about Seqera Kit. So we have a launchpad already set up here, but how did we get here? So we have a repo that is an example for y'all on those. And we can go through and use Seqera Kit here, which is just a nice templating CLI that then takes YAML files like this for the compute environments and allows you to configure all of it in code and check that into a repo. And then you can GitOps the entire workflow, which is really nice when you're working with a team and you're wondering how did this get created or recreating a experiment with an environment. So maybe not so much with the compute IMS, but maybe more importantly for, say, the Launchpad, and where you could have a couple of different pipelines and you want to update them piecemeal and change them out or change certain parameters, etc. based on those and based on your team. So it allows kind of one bioinformatician to really take a wide span of all these things. So there's a couple different pieces to this. We have our teams, as we saw earlier, with participants, and we have workspaces for the demo, for example, on each of these. And you can specify all that out outside of just click-opsing the platform. So that's kind of one piece of automation that we encourage here as well. So let's take a look at this launch. So we have the workspace, the pipeline, which is going to be Seric, which is a whole genome sequencing pipeline from NF Core. And then we have the out directory that we expect that to do. And then so we can go in here to the actual platform. And so this is something that you can configure for the wet lab members of your lab, of your organization. It's pretty powerful for the bioinformatician to be able to create these launch pads and then go and hand them off to other members of the org, and they can start to self-serve. And suddenly they're not waiting on you to launch these jobs and kick them off and get to it. They can start to do it themselves. So we can go ahead and launch here on this. And so that'll take them instantly to the run parameters, if it'll load for me. But we can go back here first to the general config. So this is just what pipeline they want to launch. These are various configs that you can specify. You can lock down the revision number. You can change it to just a commit ID as well. You can give various labels to it and say like launchpad. So let's just say this is coming from the launchpad, for example. You can specify a compute environment ahead of time in the work directory, things that the scientists may not need to think of. But then you can get this nice set of parameters and go through and take a look, and they can go through and self-serve on each of these and all the various different toggles that they can change in the pipeline. And you can set this up with a schema with NF schema for that. And so you can specify an out directory. They can make this like their bucket, for example, of automation and give that, and they can change it to their input, and they can even browse S3 for those or upload that to a data set here. So this is data sets, and then we have the data explorer as well on those. So again, lots of various things that they can start to self-serve on those. You can just build the pipeline. Suddenly you're focused more on the code and you're having meetings about talking about, hey, this isn't working with the pipeline the way I expected, instead of, hey, can you run this thing for me on it? And it really starts to speed things up, and it makes everybody a little bit happier overall and less adversarial and more of a team aspect on those. There's also advanced settings, but these are again things that the user doesn't need to do. So at any point they can just click launch here and kick off the job from that. And that takes a moment because it's got to go talk to AWS and come back and all of those things. So we'll jump over to the runs page here, where you can see we've got a couple of runs that we did. So this would be one, and maybe so if we go back, now it's launched. So this is our Launchpad job, and you can see that it's submitted and kicked off by me and each of those. So then you can hop into the Seric one that we've already done. You can see we have 23 jobs succeeded. You can see the runtime. You can also see the estimated cost, which is a very important piece when you're doing cloud computing on these. And you can start to really piece apart how efficient your workflow is. So this is a test data set. We spun it up with very minimal CPUs. So not accurate here, but you can start to see the CPU usage, the physical memory usage, how long your tasks take. You can start to identify bottlenecks in your Nextflow workflow by using platform on those. So another piece that I like to show as well is the task details. So when you're running in the cloud and you start to use Nextflow there, usually you're used to either going from local and then to an HPC environment where you kind of have access to all the files, but then suddenly in the cloud, you're using computers that just spin up for a second and then they're gone and the files are gone and you can't go debug these things. And that's where platform really comes in and starts to enable this in the cloud. So you can see all the duration and the containers and all the various like pieces of that. And that's not too difficult, but you can actually go and get the execution log and then that's saved. So you can see how much of the time on that. And then my favorite part is then you can hop into Data Explorer and you can start to look at these files actually and download them directly from here. So I could click Fusion Log and then it'll download, for example. And so that's really useful when you're going in and debugging these automated jobs, because sometimes that can be really hard when you set up these systems in the cloud and it's difficult to hop in and figure out what went wrong. So this is the actual work directory of that specific task. So when you're reviewing these, you can see parameters, configuration. You can see what data sets may have been used in that, the full execution log as well for platform. But let's also talk about reports for a moment on those. And I think this is a really important part for users to go in and self-serve as you're building these automation tools. So we're going to pull out this multi-QC report and just preview this. So they can not only kick off the jobs in platform that you've set up for them, they can then walk in here and start to self-serve with the various reports and then take them and present them across the organization without you having to download them. Once again, test data set, so ignore the failures, but this is multi-QC. If you haven't heard about it, it basically like brings all of your different data sources in from your workflow. So you have like fast QC, you have your GATK4, mark duplicates, all kinds of tools. I think it has over like 120 at least on those. So you can also just click download and they can download these reports directly from that. You can configure that to be any type of report if you have a internal like Cordo dashboard or some other thing that gets built at the end of your presentation or at the end of your pipeline on those. So that has kind of been the automation around Launchpad and platform. We also talked a little bit about Seqera Kit on those. Now, let's talk a little bit about actually automating these. So the situation that I think a lot of people are familiar with is having a sequencer, and then after the sequencer is done, it's ran for, 24 hours or so, and then it kicks off a job and it's finished, but you don't want to have to take those files then and then manually launch a job from that. You want to automate that. So we can hop into the Actions tab here on those. And so this is just a nice way that we have an HTTP endpoint created for you for for a various job that you would want to kick off. So you can see if we click Add Action here, you can see all the various things you can configure. We have compute environment and the pipeline that you want to launch, revision number. Again, very similar things. You can also have a resource label. So this might be in that. And then you could also say like the source is automation is another one on those. And those will get propagated to your AWS pieces as well. So you can manage costs better internally and see like how many of these are automated, how many of these are people kicking things off and things failing. So going back to the example action here that I've configured, we have an API for Seqera, and then we have the actions, and then we have the actual action ID and the workspace that we want to use. And so this is just a quick little curl example, but we've kind of taken that to a step further. So now we're going to hop out of that world and we're going to hop into the temporal world on this. So as Ken told you, temporal is a great tool for managing kind of this business logic, kind of like AWS Lambdas, but maybe a little more jointed and less one-off and kind of like launching something and just leaving it. This is kind of managing the business logic behind that and kicking those off. So I've just kind of had this running in the background the whole time. And so this is just a temporal workflow that is monitoring the sequencing directory. So this is the directory that's monitoring here. And I'll just close that to avoid any confusion. So you can see here in our activities in the code, we are hitting that exact same endpoint here based on those. So based on this actions, and this is a lot of Python, but you can see that we're hitting the actions space here and we're launching it for those and passing each of those in with the various parameters. This is just one way of doing it. There's tons of different ways. We have examples for just plain Python as well on those. You can kind of do it however you want to do it. So let's kind of hop into this and let's pretend in our sequencer, in our example, we have a sequenced file. So it's saying that like we finished sequencing. Now you can do something with it and trigger it. It's a common workflow for sequencers to be like, hey, this is how I'm done. Because in bioinformatics, we love files. So I'm just going to drop this in here and say it's done sequencing, and that will kick off the temporal workflow over here as this refreshes. But you can see that now we have a processed file that says like, okay, I finished this, don't keep checking this directory. And then we also have created a CSV here. And so this is going to be our sample sheet for Seric. And that has all the paths there. So over back here in temporal, we've been monitoring and we've been watching that workflow, but now we have a separate workflow to kind of manage the business logic of this, and it has a fetch metadata, and then it uploads the data to S3. So we've first fetched all the metadata, created this sample sheet, then we've uploaded all of these files in this directory to S3, and then we've triggered a workflow here. And so now it's just waiting for the workflow to finish and kick off on those. So you can see that this is our input and our results there on each of those. So if we go back to platform over here, we can go to the runs, and you can see now that we have a brand new job for Nextflow. And this is kind of about using the right tool for the job on that. So we've kicked that off, and you can see that the source from that one was the automation with temporal, not from the launchpad on those. So from that, that's kind of the entirety of the temporal workflows on those. Again, it's a great way of creating the business logic around that and managing some of these external services versus doing that all in Nextflow, and where you can kind of do your scientific logic in Nextflow and write all of those workflows to manage those pieces. And then you can manage all of these external services through something like temporal and kind of space all of these out. And it can handle maybe more of the start and stop of those versus Nextflow is meant to be a pipeline that's ran end to end and maybe resuming versus handling a ton of failures from external services and hitting API endpoints. And so from that, we can hop into the data explorer is kind of the next piece. So this is kind of after our pipeline is finished and our automation is finished. Now our scientists want to go back in and we want to start to look for these. We want to start to actually look at our results. So we have a SLAS 2025 bucket that we've been writing to for event-driven bioinformatics. And so from that, you can see all of our results. You can see the CSVs that we've uploaded. You can see the Hello Automation here on these, and we'll just hop into this directory, and you can then hop in and see our various reports like I was talking about based on FastQC. But more importantly, you can, and this is just coming off of a fresh run, so we can hop into this results, and then you can see our variant calling, and we can start to download all of these VCFs from each of these and the various paths. So that has been Data Explorer. Let's also touch on Data Studios as well. So this is the next tab over here, and you can kind of see that we've come full circle on the entire platform. So this is our latest offering. It has various different pieces. So this is where you can allocate certain CPUs to it and memory. And then you can also go in here and mount the data just to kind of give a reference here first, and then we'll hop to it, and it might make a little more sense in a moment. So you can go in here and use Fusion in the back end to mount all the data. And I don't think I've touched on Fusion yet. So behind the scenes, all of this is kind of fueled by a Fuse file system that turns S3 into an actual file system that you can use within these Nextflow workflows. That allows you, what that means for you is that allows you to keep the storage really cheap by using just S3 instead of EBS or some other fancy file system. And then you can also, but at the same time, it keeps the workflows fast, because if you buy all of these CPUs and you have this bottleneck of just using S3 to either stage your files in and stage them out, it really slows down the whole thing and you end up spending more money on it versus you can save a lot of money with Fusion on those. So that's what is powering Data Studios here. And so you can then hop in and launch various pre-made Docker images. You can also build your own container as well on those. You can give it a name, and you can also just install packages like you can with Seqera containers on those. And so you can just click next, and then you can see the overarching view of that, and you can click add and start on those, and it'll launch that, and it'll start creating those. So that's something to keep an eye out for. And I believe we are also having a webinar next month on that. So sign up for that if you're interested more in data studios and the offerings that we have on that. And so with that, I will turn it back over to Ken and we'll do some Q&A.
Thank you, Edmund. So just to kind of remind kind of what we went through, Edmund showcased how Seqera platform, Temporal, and Nextflow can automate the entire process of uploading data, fetching the metadata, triggering the workflow, doing the monitoring and logging, generating the report, and then doing additional exploration and analysis. And that really helps in terms of like being able to replace as many of those human fallible links as possible in the flow of scientific data generation and analysis. So in terms of next things, we did have a couple. Oh, we wanted to call out that all of the code that we showcased and used in our demo today can be found at Seqera Labs/Hello Automation. So if you want to see an example of that, we have a pretty simple example put together that's focusing on just kind of like showcasing how Temporal and Seqera interact, but it can definitely be built on and made into a much more complex and robust and resilient architecture that can handle different types of failures. So we're going to put up a quick one question poll for anyone who's interested in knowing more about Seqera. Feel free to go ahead and respond to that. And if you are interested in knowing more, we have a couple of great resources, one that is currently out and one that is coming soon that you might want to take a look at. There's a blog post on automating resource creation with Seqera Kit that can dive into more detail about how to use Seqera Kit to sort of programmatically control your infrastructure. And then our next webinar for next month is going to be a deep dive into studios and talking about just how to go from that pipeline to interactive analysis. And we'll showcase examples of how that can enable scientists to work more efficiently and spending less time fighting with their infrastructure. Another thing to call out, the Nextflow Summit by Seqera is going to be happening in 2 months. This is the last week that the early access bonus, you can get 25% off registration if you register by the end of this week. And it's really going to be a fantastic event with talks by both focused on science and organizational impact and bringing together all of the people who really care deeply about Nextflow and reproducible bioinformatics. So let's move on to the Q&A session. If you have any questions, we'll start tackling those in the Q&A. So one question about self-hosting Temporal on top of Kubernetes. Yes, Temporal is an open source product, and it consists of kind of 2 main pieces. There are the workers, which are the ones that are going to be picking up the tasks and working on processing them. And each worker, depending on where it's running, can be assigned different types of tasks that it's allowed to work on. And then there's also the Temporal server. Edmund was running a development instance of Temporal just on his local machine. But it can also be self-hosted in Kubernetes in a more resilient fashion. And then Temporal, the company, also has a cloud commercial offering that can also be used. So any other questions about temporal or other parts of the automation that we've talked about today? Okay. Um, question. Can you trigger on tickets, for example, from Jira? Ooh, that's an interesting idea. Um, I think that definitely should be possible to do because let's say, for example, like Jira definitely has like a robust API that you can tap into. And so temporal could interact with Jira, for example, let's say pulling Jira like every 10 minutes or so to see if there is a new ticket that has been opened within a certain project. And then it could fetch the metadata that is required in order to then communicate with Seqera platform and say, oh, there's a new Jira ticket that has been opened. Go ahead and start a workflow with those processes. And then in our automation demo, we also had the logic that was written into it to continue checking if a particular workflow has finished. So then you can go one step further, and when the workflow has finished in Seqera platform, you could then go back to Jira and update the Jira ticket with the data that was requested, and go ahead and close the ticket. So that's like a really good example of how like temporal kind of works well at tying the business logic to something that can handle like Seqera platform that can really handle like the scientific workflow as well.
Yeah, great answer. I think exactly. You can kind of take your current processes and maybe abstract that out with an API and say people are submitting Jira tickets for a run to be processed. You can just take that from the API in the fields, and then you can just feed it directly into your Seqera automation on those. Okay. Yeah, exactly. I was going to answer that one as well, Ken. So we have a question for, I can't find the help automation inside of cloud.seqera.io. Am I in the wrong place? You're not. So you don't have access to that org and the actual repo. You can sign up for your own account and you can reproduce that with SeqeraKit on it from our repo there and recreate the whole environment yourself. You will need an AWS account for that, however. But reach out if you have any troubles on that, and we can help you. Okay, next question there. Can we integrate temporal with N8N? I think that's how you pronounce it. I've seen that around as well. Um, it looks like from a cursory glance that you can't, like you could kick off a temporal workflow. This kind of looks like a separate workflow manager from temporal in general. Um, on those and maybe a little less like temporal is maybe a little more code heavy versus this may be a little bit more drag and drop, which is nice. You can definitely integrate Seqera platform with N8N is what I would say. I'm guessing that it looks like it supports webhooks and we support webhooks for those. So that's a great question. Um, yeah, I'd be excited to see that automation if anyone has that.
Hey, any more questions? Okay. Thank you very much, everyone, for coming today. Um, we hope this was useful, and we will be sharing out the recording of this webinar so you can share it with teammates or other folks who might be interested in the coming days. Have a great day.
Thanks.